Okay, now students, I think those who wish to join, they have joined. First, we will be discussing autoimmunity. We have already discussed it a bit in our class. Yes, good morning. Good morning to all of you. People are still joining. Let me share the PPT. Good. I hope that the slide is visible. For today, first we will be discussing the autoimmune disorder. As you are aware that the main feature of autoimmunity is that the breakdown of immune system, our body's ability to discriminate between self and non-self antigen. I have told you when B cell and T cell maturation occurs, the self antigens which are there in our body should not be targeted. That is why during B cell and T cell maturation, the antibodies which can react with the self antigen are eliminated by negative selection. Once negative selection is achieved, then we go for positive selection. I think you can recall. So you know what happens, the condition in which someone wants to join. The condition in which structural and functional damage is produced by the action of immunologically competent cellular antibodies against the normal self antigens of our body is known as autoimmunity. So you are aware that it is nothing but the development of intolerance of the antigens on its own cell. It is highly unfortunate, but it happens, and I have told you the reasons. We will be discussing it further. This type of body response results in a disease state characterized by a specific antibody or cell mediated immune response against body's own tissue. So we can say that autoimmunity is the breakdown of mechanism which is responsible for cell tolerance and the induction of immune response against the components of self. So I think if you look into this slide, all of you are aware that our immune system is grossly subdivided into two domains, innate and acquired. And acquired is further subdivided into cell mediated and humoral branch. All the branches are associated with autoimmune disorder. So you can see that in this picture we have already discussed that in a normal person's body there should never be any destruction of self antigens. Unfortunately, a person who is having those abnormal self antibodies which detect self antigens and harm those self antigens, in them autoimmunity develops. This gentleman, Paul Ehrlich, for the first time observed this and he named it as horror autotoxicus, means horror of self toxicity. This great scientist, German scientist, immunologist Paul Ehrlich, in the late 1890s, discovered this or understood for the first time the process of autoimmunity. As I mentioned, students, autoimmunity and the development of autoimmune disorders can be resulted from various factors, starting from the genetic problem, environmental induced problem, and immune regulation regulated problem. You can see over here, students, apart from the self non-self question, there are certain other reasons why autoimmunity can develop. You see, these are known as the sequestered antigen. Certain self antigens are present in the closed system and they are not accessible to the immune apparatus. They are not accessible to the immune apparatus. These antigens are called sequestered antigen. One example you can see, some antigens they remain hidden in our CNS, central nervous system, in our testes, in the cornea region of the eye. Until and unless these antigens are exposed to the blood circulatory system or lymph system, no antibody attacks them. Due to organ damage, due to wound, it may happen so these hidden antigens are released, and these hidden antigens reaches the bloodstream, then encounters the antigen sensitive cells and causes autoimmunity. So you can understand, apart from this self non-self, this sequestering of the antigens and the breakdown of the system, damage to the system, and when the antigens become exposed to the blood circulatory system, it may also give rise to autoimmune disorder. There are many environmental factors such as the ultraviolet radiation, smoking. This also causes some heightened response of autoimmune disorder. One point that you should keep it in mind, the strong gender bias is observed in case of autoimmune disorder. You can understand women are more prone to the autoimmune disorder because of their hormonal regulation, and also because in after puberty each month the menstrual cycle occurs, and there is a cycle of hormonal upregulation and downregulation of LH, FSH, estrogen, progesterone, that makes them quite susceptible to few autoimmune disorders. Now, students, you can see we will be discussing two representative diseases of autoimmune disorders. There are many, but to give you the idea, we will be discussing few basic autoimmune disorders. Okay, so one of the important autoimmune disorder is rheumatoid arthritis. You can understand it is a common autoimmune disorder. Most often it affects the human between 40 to 60 years age, and you see how it happens and why it happens. So what happens over here is that many individuals with rheumatoid arthritis produce a group of autoantibodies which are known as rheumatoid factors. They react with the FC region of the IgG. And another thing is that the classic rheumatoid factor is an IgM antibody with that reactivity. So you can understand these autoantibodies can bind with the normally circulating IgG, and by this they form the IgM IgE complex, and this IgE IgM complex gets deposited into the joints. So you can see this IgM IgG complex activates the complement cascade, and it stimulates the type 3 hypersensitivity reaction. So you can understand, and this leads to the chronic inflammation. So again, I want to make one point very, very clear, student, that please keep it in mind that these are all related with each other. Whenever you are studying, please study hypersensitivity very, very diligently, other and complement pathway, I should say. If you are not go through those areas diligently, you will never be able to understand this autoimmunity and all these parts. Now you can see in the left panel, you can see in the normal condition, in the normal joint, and the right one, the swollen, inflated, inflamed uh, synovial membrane joint, where the IgM IgG complex has been deposited. Then comes the another type of antibody autoimmune disease, myasthenia gravis. It has a different mechanism. It is mediated by blocking of the antibodies. A patient with this disease produces autoantibodies that bind to the acetylcholine receptors at the motor end plates of the muscle. So in the neuromuscular junction, these autoantibodies just all what I can say, it blocks the acetylcholine receptors. Thus, by doing so, it prevents the flow of acetylcholine in the neuromuscular junction. And you can understand these antibodies destroy the cells bearing the acetylcholine receptors. And as a result of it, the neural network, the neurotransmission comes to an halt. And as a result of it, it causes severe diseases. Without any treatment, progressive weakening of the muscle leads to severe impairment of eating, other movements. But if it is treated, it can be not totally cured, but its extent can be reduced. And you can see over here in the picture, in the normal circumstances, you see in the neuromuscular junction, acetylcholine is doing its work. But in the right panel, you can see under the condition in which myasthenia gravis is occurring, the receptors are already occupied by the acetylcholine anti-antibodies. Hence, the acetylcholine cannot bind to the receptors and no neural transmission can occur properly, and it is causing many problems which we have already discussed. So there are many treatments available. I have mentioned few over here. Please go through it. There is nothing to understand more. So we have discussed first today the first chapter, the first area, that is the autoimmune disorder. Now I will be discussing next topic. There are three topics which were yet to be discussed. One is autoimmunity, number two is tolerance, number three is KIT. So I will be discussing tolerance now, then I will go to SKIT, and then I will go to the RTD, the areas. So just a minute. I hope that it is visible. Is it visible, students? দেখা যাচ্ছে স্লাইডটা? Please confirm.
Yes, sir. Yes,
Yes,
sir.
sir.
So now we are going to discuss immunologic tolerance. The four chapters are associated with each other: complement, hypersensitivity, autoimmunity, and tolerance. We have already discussed first three. We are going to discuss the final one. So I think by now all of you are aware this what is immunologic tolerance, the state in which the individual is capable of developing an immune response to a specific antigen. State in which the individual is capable of developing an immune response to a specific antigen. What is self tolerance? It refers to the lack of individual's antigen. So you can see over here, I think already you are aware of the self and non self and all this. So you know there are certain terms that you have to understand. The first is that the tolerogens or tolerogenic antigens, the antigen that induce tolerance. And next comes self tolerance. You are aware we are all have the self tolerance. That is why we tolerate self antigen. Those who do not have the self tolerance, they develop autoimmunity. Now, there was an experiment, great experiment. I have discussed Tonigawa's bombshell in VDJ joining. Now, I am going to discuss the great experiment given by Barnett and Medawar. Please try to understand what they have done. This experiment is known as the neonatal tolerance experiment. Please look at the scheme. You can see there are three mouse: week 0, week 6, and week 7. In the week 0, in the neonatal mouse, inject neonatal mouse strain A with strain B bone marrow. We have just mixed the two type of bone marrows. In week 6, you see graft skin for strain B and strain C. And in week 7, strain B graph is accepted, strain C graph is rejected. So what does it mean? It means that the mouse over the period of time has acquired tolerance towards the strain B. So that is why it has accepted strain B's graft, but it has failed to accept the strain C's graft. So students, now we will be discussing another term which is known as ignorance. It has been shown that there are T cells and B cells which are specific for autoantigen present in our circulation. There are few. These cells are quite capable of making a response, but they are unaware of the presence of their autoantigen. It is quite unusual সেলফ অ্যান্টিজেনকে ডিস্ট্রয় করতে, অ্যাটাক করতে, কিন্তু বলা হচ্ছে দে আর আনঅ্যাওয়্যার অফ দিস প্রসেস। হাউ অ্যান্ড হোয়াই? So keep it in mind, there are two reasons for this. The first is that the antigen may simply be present in too low concentration. এতটাই কম কনসেন্ট্রেশনে আছে যে ওটাকে সেন্স করা যাচ্ছে না। দিস ইজ দ্য ফার্স্ট রিজন। And the second possibility is most interesting one, আমি তোমাদের এটা পড়ালাম অটো অ্যান্টিজেনে, সেই জিনিসটাই রিলেট করার চেষ্টা করো। Some antigens are sequestered from the immune system in the locations where they are not freely exposed to surveillance. নির্দিষ্ট কতগুলো জায়গায় এগুলো লুকিয়ে রাখা হয়েছে। They are not exposed to the circulation. So these are termed as immunologically privileged sites. I have already given you the example. Our eye, the cornea region, central nervous system, CNS, testes, these are the areas. So you can understand pathologically mediated disruption of these privileged sites may expose the sequestered antigens, leading to an autoimmune response.
স্যার সিকুয়েস্টেড অ্যান্টিজেন্স তার মানে সেইগুলো যাদের অ্যান্টিজেন প্রেজেন্টেশন হচ্ছে না বা এপিসিস?
যেগুলো অ্যান্টিজেন প্রেজেন্টেশন হচ্ছে না, কেন হচ্ছে না? কারণ এগুলো কখনোই এক্সপোজড হচ্ছে না ইমিউন সিস্টেমের সার্কুলেশনে, এগুলো ইমিউনোলজিক্যালি প্রিভিলেজ সাইটসে লুকিয়ে রাখা আছে। আনফর্চুনেটলি যদি ডিউ টু উন্ড ড্যামেজ অর আদার ডিজিজ কন্ডিশন ওখান থেকে এগুলো বেরিয়ে আসে এবং এক্সপোজ হয়, তাহলে কিন্তু অটো ইমিউন ডিসঅর্ডার হতে পারে। বলো অনির্বাণ, বলো। হ্যাঁ, যদি আমাদের শরীরে সেই অ্যান্টিজেনগুলোর এগেনস্টে অ্যান্টিবডি থেকে থাকে তাহলে হার্মফুল হবে, আর যদি না থেকে থাকে তাহলে তো আমরা কিছু এটার এগেনস্টে তৈরি করতেই পারবো না, তাহলে তো হার্মফুল হওয়ার প্রশ্ন নেই। যাদের অটো ইমিউনিটি আছে তাদের পক্ষে নিঃসন্দেহে এটা হার্মফুল হবে। সো এবার আমরা জানবো, মনে রাখবে ইমিউনোলজিক টলারেন্সকে দুটো ভাগে ভাগ করা হয়। একটা হচ্ছে সেন্ট্রাল টলারেন্স, আরেকটা হচ্ছে পেরিফেরাল টলারেন্স। দু রকমের টলারেন্স হয়। এবার আমরা এটা বোঝার চেষ্টা করি। সেন্ট্রাল টলারেন্সটা কি জিনিস দেখো, refers to death or deletion of self-reactive T cell or B lymphocyte clones during their maturation in the central lymphoid organ। যেটা আমি বারবার বলছি যে টি সেলের ক্ষেত্রে থাইমাসে অ্যান্ড বি সেলের ক্ষেত্রে বোন ম্যারোতে ম্যাচুরেশনের সময় সেলফ রিয়াক্টিং টি সেল অ্যান্ড বি সেলগুলো ডিস্ট্রয় করে দেওয়া হচ্ছে। এটা তোমরা প্রত্যেকে জেনেছো। আর পরবর্তী ক্ষেত্রে যে জিনিসটা আসছে খুব গুরুত্বপূর্ণ, পেরিফেরাল টলারেন্স। ধরে ভাবো মাসের নেগেটিভ সিলেকশনটা হয়েছে, সেটা যদি এক্স মানে সেটা এড়িয়ে গেল, escape করে গেল, then what happens? It can cause issue unless they are detected or muzzled into the peripheral tissues. So মনে রাখবে, সেন্ট্রাল টলারেন্স ফেল করলে তখন পেরিফেরাল টলারেন্স কাম ইনটু বিং অ্যান্ড দে স্টার্ট ডুইং দেয়ার অ্যাক্টিভিটি। কারণ কোনোভাবে সেন্ট্রাল টলারেন্সে জিনিসগুলো টেক কেয়ার হয়ে গেলে পেরিফেরাল টলারেন্সের আর প্রয়োজন হয় না। কিন্তু যদি সেন্ট্রাল টলারেন্স কোনোভাবে ফেল করে যায় তখন পেরিফেরাল টলারেন্সটা অ্যাক্টিভ হয়। সো কিপ ইট ইন মাইন্ড, দেয়ার আর সার্টেন মেকানিজম বাই হুইচ আমরা পেরিফেরাল টলারেন্সের বা টলারেন্সটাকে মেক শিওর করি যাতে কাজ করে। 1 নম্বর, ক্লোনাল ডিলিশন বাই অ্যাক্টিভেশন অফ ইনডিউস সেল ডেথ, অ্যাপোপ্টোসিস করে সেলগুলোকে মেরে দেওয়া। ক্লোনাল এনার্জি অ্যান্ড পেরিফেরাল সাপ্রেশন বাই টি সেল। এই জায়গাটা আমরা এবার একটু বোঝার চেষ্টা করি। ফার্স্টেই আসছে ক্লোনাল ডিলিশন। সোজা কথা, আমার উদ্দেশ্য হবে সেলফ রিঅ্যাক্টিং টি সেল বা বি সেলগুলোকে ডিস্ট্রয় করা। তাহলে কি বলছে দেখো, functionally immature cells of a clone encountering antigen undergo programmed cell death as autoreactive T cell are eliminated in the thymus following interaction with self antigen during differentiation. Likewise, differentiating early B cells become tolerant when they encounter cell associated or soluble self antigen. So keep it in mind, clonal deletion মানে যে সেলগুলো টি সেলই হোক বা বি সেলই হোক সেলফ অ্যান্টিজেনকে অ্যাটাক করছে তাদেরকে অ্যাপোপ্টোসিস করে প্রোগ্রাম সেল ডেথ করে মেরে দেওয়া। সো বুঝতেই পারছো, এই ছবিটা লক্ষ্য করো প্রত্যেকে, এটা হচ্ছে একটা প্রিলিমিনারি বি সেল, ইম্যাচিওর। সেখান থেকে যে সেলগুলো সেলফ রিঅ্যাক্টিভ হয় সেই সেলগুলোকে অ্যাপোপ্টোসিস করে ডেথ করে দেওয়া হয়, মেরে দেওয়া হয়। আর যে সেলগুলো নন রিঅ্যাক্টিভ হয় সেগুলো সার্ভাইভ করে, সেগুলো ম্যাচিওর টি সেলের দিকে, বি সেলের দিকে চলে যায়। তো তোমরা নেক্রোসিস আর অ্যাপোপ্টোসিস আমি তোমাদের আগেই পড়েছি যে নেক্রোসিসের ক্ষেত্রে টিস্যু ইনজুরি, কোনো ড্রাগের এফেক্টে কোনো কিছুতে সেলটা যখন ব্লিডিং আউট করে ডিস্ট্রয় হয়ে যায়। আর অ্যাপোপ্টোসিস প্রোগ্রামড ওয়েতে ধীরে ধীরে স্টেপ ওয়াইজ সেলটাকে ডেভেলপ মেরে ফেলা হয়। এবার যে জিনিসটা এর পরবর্তী জিনিসটায় আসছে তার নাম হচ্ছে ক্লোনাল এনার্জি। দেখো, অটোরিয়াক্টিভ টি সেল when exposed to antigenic peptide which do not possess co-stimulatory molecule become allergic to antigen. B cells when exposed to large amount of soluble antigen downregulate their surface IgM and become allergic. তাহলে মনে রাখবে স্টুডেন্টস, যদি এমন হয় কিছু টি সেল বা বি সেল যেগুলো কিন্তু অ্যাক্টিভ হওয়া উচিত ছিল না কিন্তু তারা সেলফ রিঅ্যাক্টিভ এবং অ্যাক্টিভ হয়ে যাচ্ছে, অনেক ক্ষেত্রে তাদের অ্যাক্টিভিটিটাকে রেডিউস করে দেওয়া হয়। তাদের যে কোস্টিমুলেটরি মলিকিউলস, সেই কোস্টিমুলেটরি মলিকিউলসের অ্যাক্টিভিটি কমিয়ে অথবা সারফেস আইজিএম আইজিজির অ্যাক্টিভিটি কমিয়ে। সো বাই দিস প্রসেস ওয়েন সেলফ রিঅ্যাক্টিভিটি ইজ রেডিউস, দিস ইজ নোন অ্যাজ দ্য ক্লোনাল এনার্জি। এবার লাস্টলি যে জিনিসটা আমরা তোমাদেরকে বলবো সেটা হচ্ছে পেরিফেরাল স্টিমুলেশন অফ টি সেল। ফাইনালি, বেশ কিছু ক্ষেত্রে কিছু সাপ্রেসার টি সেলস have the ability to downregulate the function of other autoreactive T cell. So কিছু সাপ্রেসার টি সেল এমন কাজ করে জায়গাটা পড়লাম, নিজেদের পড়তে হবে স্বাভাবিকভাবে যে টলারেন্স মেইনলি দু রকমের হয়, সেন্ট্রাল অ্যান্ড পেরিফেরাল। সেন্ট্রাল টলারেন্স ফেল করলে পেরিফেরাল টলারেন্স অ্যাক্টিভেট হয় এবং মেইনলি তিনটে মেকানিজমের সাহায্যে অটো রিঅ্যাক্টিভ, সেলফ রিঅ্যাক্টিভ যে অ্যান্টিবডি, যে বিসেল টিসেল সেগুলোকে ডিস্ট্রয় করা হয়। ক্লোনাল ডিলিশন, এনার্জি অ্যান্ড টিসেল সাপ্রেশন। এবার আমরা নেক্সট যে জিনিসটা স্কিট, সে জায়গাটা পড়া স্টার্ট করব। জাস্ট I hope that it is visible. So আমরা এবার করব combined immunodeficiencies. So combined immunodeficiencies or combined immunity deficiency, these are the immunodeficiency disorder that involve multiple components of immune system. It includes both the humoral as well as cell mediated immunity. So তোমরা প্রত্যেকে জানো mutation of a particular gene may lead to split or milder combined immunodeficiency depending upon whether the gene defect is fully penetrant or on the functional consequences of the specific mutation. It may happen a mutation may happen in a particular gene and terms, the two terms I think we can recall penetrance and expressivity. Penetrance means a genotype, how much is manifested in a population, a particular trait. And expressivity means how much it is expressed in a particular person. Please, students, I would request you, whenever you are studying immunology, you have to have a very good idea about these two terms of molecular biology, that is penetrance and expressivity. So skit, one of the terms which is associated with skit, you can understand, unfortunately in this skit, that is severe combined immunodeficiency, neither the T cells nor the B cells work properly. Babies with skit can't produce IgG. So once the IgG from the mother has gone, they easily get into the types of infection. Unfortunately, they die very soon. So you can understand a baby with skit can hardly survive because without IgG it is quite impossible to survive. So now let me introduce one term which is known as the bubble boy disease. It is often called as this bubble boy disease because it was widely known in the 1970s and 80s when the world learned about David Vetter, a boy who had X-linked skit. The mutation was associated with X-linked. Unfortunately, he died at the age of 12, but till that, in a big bubble, in a big plastic bubble, germ-free bubble, he was kept there. So you can see this is why it is known as the bubble boy disease. There are many types of skit. One of them is known as the leaky skit. It is due to the incomplete mutation. So what happens? They produce some extent T cells, B cells, but not to that extent that they are normal. Some are called the variant split. And you can understand depending on the type of mutation and where the mutation is happening, there is X-linked, autosomal, different kind of splits are present. So you can understand in molecular biology research and in immunological research, the importance of speed mice is very, very important. So you see, in 1983, Melvin and Gail Bosma and their colleagues first time they described this autosomal mutation in a skid mice, and skid mice was developed in the lab. So it is a technically molecular biology term. It is used in the molecular biology lab where intentionally mutation of certain genes are done and a mouse model is produced. The mouse in which there is the mouse in which there is huge mutation, and people study each mutation and try to understand how it is associated with several other diseases. So you can see there are different kind of squid mouse models available in which different genes are mutated. So you can understand some of the examples of some diseases. One of them are X kit is given over here. Please go through it. This is very easy. This is very important. Adenosine deaminase deficiency. Again, this catalyzes the conversion of adenosine into inosine, and its deficiency leads to the accumulation of adenosine, and which has a negative impact on the DNA synthesis. So please keep it in mind. There are many kinds of speech in in your syllabus. I have mentioned few. Please go through it. This is very simple, and if you have any query, please feel free to ask. So with this, your syllabus in immunology comes to an end. From my part, I will be sending you the worksheet which you have to go through, and you have to make sure the questions mentioned in the worksheet you are aware, you are able to do. So now we will take about it's 10 6 2 minutes break. Then I will be starting the two three topics of art data. So just take a break for 2 minutes and then I will start.